Welcome to Tensor Lens. Today we're going deep on a system that hacks the human eye at the single cell level. It generates a sensory input that literally does not exist in the natural world. And the result? It allows someone to see a color that, until now, was biologically impossible. Let's break down the engineering.
So, let's start from first principles with a simple question. Is the color green actually real? Now, if your mental model is that green is some physical property just sitting on the surface of an object, well, that model is flawed. The physical reality is way more complex, and honestly, it's a lot cooler. Color isn't out there in the world, it's a calculation. It's something your brain builds.
You see, your entire experience of color is a computational construct. It's your brain, in real time, decoding signals from just 3 types of photoreceptor cells in your retina. These are the L, M, and S cones, basically your red, green, and blue sensors. What you call green is just your brain's interpretation of a specific firing pattern, usually when your green and your red cones get stimulated at the same time.
Okay, but this whole system, this way of seeing the world, it actually creates a fundamental problem. It's a biological lock that has put a hard limit on what humans have been able to see forever. Here's the catch. Because of the way our cone cells are designed, their sensitivities overlap. Think of it like their signals kind of bleed into each other. This means that no single wavelength of light that you'd ever find in nature can hit only the M cones, your green sensors. Any green light from the sun or a plant also triggers the red cones and even the blue ones a little bit. A pure, clean signal to just the green cone, it just doesn't happen.
And that creates what you could call a forbidden sensory input. It's a combination of signals that the human brain, over its entire evolutionary history, has never once had to process. A pure, isolated M-cone signal has been locked away by biology. So you can see the engineering challenge, right? The question becomes, can you build a key to pick that lock? And that is exactly where this engineering breakthrough from UC Berkeley comes into play. They designed a system specifically to bypass that biological wall.
They call it the Aussie system, and what it does is pretty mind-blowing. It's designed to totally sidestep the messy reality of natural light and take direct programmable control over individual cone cells. It is, for all intents and purposes, a direct interface for sending the brain that one signal it was never supposed to get.
Now, the engineering to pull this off is just incredible. This is not just pointing a laser at someone's eye, it is a complex 4-stage process. First, the system uses advanced imaging to create a complete map of the subject's retina, identifying the exact location and type of every single cone. That's your target map. Second, it uses adaptive optics to cancel out the tiny, unique imperfections in the person's own lens, making the laser beam perfect. Third, it has to track the eye's constant tiny jitters, microsaccades, in real time. Only when all 3 of those things are perfect, target locked, beam focused, and motion stabilized, does the system fire a tiny pulse from a green laser, timed perfectly to hit one single M cone and nothing else.
The precision here is so extreme that the researchers themselves use the best possible analogy. They are painting colors directly onto the retina at a resolution of a single cell. Think about that. They're treating the retina like a biological high-definition display and writing information directly to its individual pixels. That is an absolutely unprecedented level of control.
So this is the moment of truth. You built this insane system. You picked the biological lock. What happens when you feed the brain a signal that it has never, ever seen before in millions of years of evolution? You get a perception that has never been experienced before. The result of zapping only the M cone is a new color. The researchers named it OLO. And if you want to describe it with data, in the coordinate space of cone activation, it's simply 0, 1, 0. That's 0 input from the red cone, 100% from the green cone, and 0 from the blue cone. That clean, impossible signal, delivered. But of course, we don't experience the world as numbers in a coordinate space. So what did it actually look like? Well, all 5 observers in the study described it in remarkably similar terms. They called it an extremely saturated blue-green, something with an intensity that was way beyond any normal green light they could compare it to. This wasn't just a new shade, it was a fundamentally new visual experience, a new quality.
And here's what really proves it's real and just how insane the precision is. This perception of OLO was incredibly fragile. It only existed with perfect laser alignment. If they nudged the beam just a tiny bit so it hit a neighboring red or blue cone, poof, the OLO perception would instantly vanish, and subjects would just see a normal spot of green.
That is the perfect control experiment, right? It confirms Olo isn't some weird system artifact. It is the brain's direct interpretation of that pure, isolated signal.
So, could you see this color on your computer screen? No way. This color is completely outside the gamut of any display we have. It's outside the gamut of anything in the natural world. When they asked the subjects to try and match it on a screen, it was basically impossible. The closest they could come was to take a super bright teal color and then wash it out by adding white. This is a huge clue that Holo Atolo isn't just on the edge of our vision. It occupies a whole new territory we could never get to before.
So, what does all this mean? I mean, this is so much more than a cool lab trick. The ability to just create a new color sensation has profound implications for neuroscience and how we think about perception itself.
Here's the core takeaway. This experiment is the first direct proof that our color space, the range of colors we can see, isn't defined by the physics of light. It's defined by the decoding software running on our neural hardware. If you can hack the input signal, you can get a brand new output from the brain. Our potential to see is demonstrably bigger than the slice of reality nature normally lets us access.
So where could this possibly go? Well, the applications are definitely a long way off, but they sound like something straight out of science fiction. You could potentially use this to simulate tetrachromacy, to see what it's like to have a fourth color cone, or maybe even simulate the vision of other animals, like bees or birds. And on the clinical side, you might one day let a colorblind person experience the colors their own biology denies them. Or you could model exactly how retinal diseases affect vision, one cell at a time.
Okay, but now it's time for the hard reality check. This is where we have to pump the brakes. Because as incredible as this research is, this technology is nowhere near ready for anything outside a very expensive, very controlled lab.
The experimental setup is intense. Subjects literally have to bite down on a metal bar to keep their head perfectly still. The effect doesn't even work in your sharp central vision. It only happens in a tiny patch of your peripheral vision, a patch only about the size of 2 full moons in the sky. And the equipment? We're talking incredibly expensive, specialized lab-grade lasers and optics. So let me be perfectly clear. This is not coming to a VR headset or your phone anytime soon.
And good science always has debate. It's important to know that not everyone is convinced Olo is a truly new hue. Vision scientist John Barbier has suggested that maybe it's not a new color category at all, but just the brain's perception of a green with a saturation level that's impossible in nature. A quantitative difference, not a qualitative one. Now, the subjective reports from the people who actually saw it seem to push back on that, but it's a valid point in the ongoing scientific conversation.
So let's just summarize the core thesis here. This research gives us direct, hard evidence that color is not a property of the world out there. It's a product of the computation happening in here. By engineering a key to break the biological lock on our senses, we've shown that the brain has this latent, hidden capacity to construct brand new perceptual realities. We don't see the world as it is. We see the model of the world our brain decodes for us.
The Oz system has opened a new door in our understanding of perception, proving that what we call reality is ultimately a matter of neural decoding. If you value this level of rigorous analysis at the intersection of engineering and neuroscience, subscribe to Tensor Lens for more deep dives.